Hi, we're going to go over mitosis and just those stages and what happens in each phase, each part of the phases. Yes, there's interphase that is actually part of the phase a normal cell will spend most of its life in. And that's the phase where, so I guess it could be resting phase. It could be an interim phase if it's going to replicate, or some cells never get out of interphase and they never replicate. So that would be where the resting cells like to sit. So maybe non-dividing cells stay there. The DNA is always unwound as opposed to wound up in those chromosomes. And doubled. I should say unwound, or sorry, unwound. Unwound and doubled. Okay, we're just going to go with it. All right, so this is organelle numbers can increase here, but what's happened is the cells received a signal to divide. That's pretty much what's going to enable us to get to the next mitosis. Okay, so cell signal, whatever it is, if there's been some damage and cells next to it need to replace the damaged cells, and a cell checks itself, has to have enough energy and molecules. So what's going to happen then is the DNA is going to start to wind if the cell has enough molecules and energy. Organelles are going to double. And DNA is already doubled because cells like to operate that way anyways, okay? All right. Let's see, resting here, we could make sure that we know it. Resting is interphase. But once it's decided to do mitosis, we also call that nuclear division. We could also call that replication. The cells replicate itself. Or multiplication. I guess it's replication, really. I guess replication and division are the same thing in biology. Okay. Then after the cell's undergone mitosis, there's two cells that are going to be created. The cytoplasm has to be divided, though. That's going to be cytokinesis, cyto meaning cell, kinesis movement. So the cell splits into two daughter cells. In this case, they're identical. Most cells undergo regular mitosis, except for weird ones. Okay, so the weird situations could be muscle cells that are going to pretty much elongate. They're not going to actually end up doing cytokinesis. They're just like connected multinucleated cells so that the muscle cell signal to contract propagates down all the cells. So they stay together. So muscle cells don't do regular mitosis. So I'll say not muscle cells. Here we go. Not muscle cells, especially the heart, by the way. For all intents and purposes, the heart doesn't really do regular mitosis. So just it actually can possibly replicate at 1% per year. So that's why having a heart attack and having a heart damage is so bad. It's not like your muscle cells of the heart are just going to repair itself at a normal rate. Also, your neurons, they don't undergo regular mitosis. They might get their axon chopped off or done right and they try to regrow it. But it's good that neurons don't do regular mitosis because otherwise we'd forget how to move. Like literally, we'd lose attachment because the neurons rely on that signal being transmitted and those attachments that have been made since before you were born. Some of these nerve cells. So neurons in the brain as well. They kind of more they rarely divide. There's some cases where there's brain damage and these guys could be signaled to divide. But if you have to say regular mitosis, muscle cells, neurons, those are not really on the list. Also, I guess red blood cells, because remember, red blood cells, they sort of spit their nucleus out when they divide. The nucleus doesn't really, I don't know. So I guess and the thing that we need to talk about is differentiation, because why all cells have all DNA, right? But not all cells act the same, and not all cells have all their DNA active. So what happens is cells become differentiated in your body. So like only muscle cells are going to make other muscle cells, not skin cells and other kinds of cells. But what makes them different? So let's write down here. Every cell has a full set of genes. Okay, every body cell, but just has certain cells or certain genes turned off. Okay. Mitosis, want to make sure you know, yeah, that only happens in body cells. And its purpose would be growth and repair. I mean, you're going to need to make some more red blood cells to replace the old red blood cells when those are kind of worn out. And with regular mitosis, you're going to begin with one cell that actually has 92 doubled chromosomes. Okay. So it has 92 chromosomes. It has that are twins. I say 92 chromosomes. So doubled DNA. And we call that a haploid number or a diploid number because you actually still have one gene from mom and one gene from dad for like eye color. So you have two sets of genes for every kind of location where there could be genes. So normally we'd have, like, if you didn't have any twins, you'd have 46 chromosomes. So 46 twins, identical twin chromosomes, will make 92. And then we're going to split those in half so that each cell gets a set of 46. So the result will be two identical daughter cells, each with 46 chromosomes. And how I remember it is in your sex cells, they only make chromosome cells with 23 chromosomes, like your half haploid. That's your haploid number. Because when you're going to make kids, they only need one eye color trait, and they only need one like bridge of the nose trait, and they only need one like freckles or no freckles, or I don't know. So 23 came from mom and 23 came from dad. 23andMe, and that's how you got your 46 chromosomes in your body cells. And yeah, so that's how I kind of remember 46. And then of course your cells like to double your DNA anyways, because yeah, that way you have extra DNA. It's great. Make more proteins, make more RNA, it's more efficient. And then your cell can divide. Okay, let's try to use rainbow colors for the mitosis cell steps. If you could print this out, that might be really helpful. It's kind of on the internet. It's just something widely available, I guess. Fairly widely available. All right. And I want you to know that the arrows are the cells going to the next stage. It doesn't have anything to do with the box. Okay, so the box for this guy here for interphase, is that the right color? Jeez, looks pink. All right, goes with this guy. I'm going to start with E because interphase is where all cells start, and also kind of where they end if they're they already exist. So some structures here to kind of recognize. Cell membrane. There's the nucleolus right there. Remember, that's what makes RNA. Okay, you can see it. It's visible because it's a lot of large chunks of RNA in one area. Okay, and then centrioles here, by the way, they like to be doubled always. We'll talk about that. There's one of those organelles that like to be doubled anyways. All right, and then wound up here is chromatin, which basically is unwound DNA. You can't really see it too well because it just looks like a grayish area, lighter gray than the nucleolus. So chromatin, and they call it chromatin because it would dye. It would make a color. Like a chrome means color. DNA is already doubled. Or we could say it's doubled in interphase. Okay. Ooh, a red. All right. What's going to happen is the cell is going to receive a signal to divide for whatever reason. Well, that's another whole ball game there. And it can divide because it has enough, and it's checked. It's got enough DNA. It's got enough ATP. It's got enough organelles, enough energy because everything takes ATP. All right. So, I made interphase red. Okay, I'm going to make the next one prophase, of course, orange. Kind of the progenitor, this beginning of mitosis. And you'll see centrioles here. Yeah, that's the centrioles. We also call them asters at this point, which just means star, because they do look like stars. When the centrioles grow, they're spindle fibers. And that's what helps them migrate. They like push each other apart, and they create poles. So they create one end of the cell and an opposite end of the cell. I don't have very skinny markers, I'm afraid. I have all fat markers. So spindle fibers are here. We talked about those, like microtubules. Notice that the nucleolus disappears. It's gone. The nucleus is disappearing too. All right, so let's kind of just mention that. And chromosomes are wound here, and they're with their twin. The DNA is doubled, so they're attached to their twin. Twin chromatids, we could say. So that's another way you can tell. I said wind, but I think I spelled wild. That's a way you can tell that you're in prophase, is that the chromosomes are visible. I don't know if we can recover from that very well. There we go. Just go with it. And nuclear membrane disappears here too. All right, so that's pretty much at this point. We already have our diploid number, and it's doubled. It's a doubled diploid number. I like to write that down just to keep in mind. See, chromosomes are already doubled. And we say diploid because we want you to know it's not like you're getting 96 chromosomes from one parent. You got, anyway. Just like to distinguish that. Metaphase, meta means middle. And I don't know, I don't think I can get away with using yellow here. But I'll take marker and then I'll just, highlight it over it. Meta kind of means middle because we are lining up the chromosomes in the middle of the cell, and that's how you can tell metaphase. I think prometaphase is where they're almost lined up, they're not quite straight, okay? Chromosomes line up in the middle. That's what characterizes it. And that's the spindle fibers that are doing that. In fact, they are attaching to each one of the centromeres of each set of chromosomes. And by the way, like their two of sets are dark because like this chromosome you got from your dad, this one you got from your mom, this one you got from your dad, this one you got from your mom, something like that. And the spindle fibers attach to each chromatid at the centromere. So here's the centromere, that black spot. And spindle fibers are attaching. Okay, and then they're going to pull back and retract in the big breakup anaphase. So I'm making that all yellow because, rainbow and stuff. Does anybody know anybody named Anna that had they were dating somebody named Anna and had a big breakup? Yeah. Or Anna could mean like against, kind of like anti, just against, they're going to go opposite directions. They're going to go, you go there, you go here. And they separate the chromosomes. I didn't highlight it all yellow. That's okay. Anaphase as opposed to gestaphase. Just kidding. Chromosomes separate. And now they're called chromatids, right? Cool. Enough said. All right. Also, the organelles are being pulled, like mitochondria has its own spindle fiber, and then they make sure that they got one on each side. Because if you don't have any mitochondria in the new cell, it's not going to survive. We don't make mitochondria. Mitochondria already exists. It has its own DNA. It replicates itself. It could be said that it was kind of possibly a earlier version of a prokaryotic cell or some sort, and it got sort of hijacked by a regular cell. I mean, probably a symbiotic relationship. There, that's better to say. And then they're like, okay, I'll give you molecules if you make energy for me. And he's like, cool. So then they stay together. There's a good mutual agreement. I don't know how many tens of billions of years ago, perhaps. All right, the last one, telo, kind of like could mean like transport, teleport, but you can remember it because it looks like maybe you have like the nuclear membranes are forming and they look like the two ends of the speaker of an old-fashioned phone. I don't think you're going to find that looks like a phone to you, but it could if you're a child of the 80s, I guess. That's terrible. Okay, the side view is like this, right? But these are the speakers and they're still attached. That's what I'm trying to say. And that's the phone cord. There, that's better. Second try, always better. Nuclear membrane starts to reform. That's why you could see, and that's what I call like the two speakers of the phone. Okay, that happens. And the nucleoli reappear. The nucleolus of each side, plural, is nucleoli. Okay. They reappear. Because you need to start doing stuff. The cell needs to do its metabolism and start making proteins. So it needs to start creating RNA, junk like that. Chromosomes begin to relax and unwind. So they start to disappear. Yes, I like it. They relax and unwind, just like we should at the end of a hard day. Or you start making videos for your online students. JK. Don't worry. I'm gonna go play some soccer. So that's cool. And spindle fibers start to disappear because they're not needed. And I believe the cytoskeleton that kind of had to disassemble and in order to enable organelles to move, the cytoskeleton starts to reappear, but we don't need the spindle fibers. But the centrioles duplicate. Okay. just in case we need to do replication. I think they said that a lot of nerve cells don't have centrioles, so that's the other reason, like that, the natural way that stops nerve cells from just from dividing. Then I guess the next part, some people call it a phase, I guess, because yeah, cytokinesis. Ooh, we could do purple. Let's fill this rainbow out. That happens in between telophase and interphase. Cell means cyto, and kinesis is movement. So the cells literally become two cells. Finally, the cytoplasm divides. And the organelles are already divided pretty much. One on one, some on side and some on the other. Okay, hopefully evenly. And you get two identical daughter cells. The DNA starts to double. Yep, because it's been unwinding in telophase. So now, once it's unwound, it can fully replicate the DNA. And of course, we have two identical daughter cells, each with the 46 chromosomes, and those are going to double to be the 92 number. Okay, and that's all she wrote. For mitosis, which happens in your toe, in your body cells, what happens if your sibling steps on your foot, you say mitosis, and then after your toe cells are damaged, your toe cells are going to do mitosis to replace those toe cells. So that's different than meiosis, which is, oh my, I have a cis, which is how your sister got created, perhaps, and you, and any human. Sure. So we'll talk about that next time. Thank you, ladies and gentlemen, for joining us. I hope you're having a great weekend or a great day, whichever day you end up watching this. So see you later.